So thank you so much for that introduction, Linda. And I really have a heartfelt thanks out here to Linda, Michael, everybody in the DARE Collaboratory, as well as Patricia, who really worked so hard to bring, to let the community know what everybody in the scientific field is doing and working towards a cure for HIV. And I'm also really grateful that you all called me to talk about a little bit of our work that we're doing with the HOPE Collaboratory in terms of silencing HIV and how we are using newer age molecules like CRISPRs or even better molecules to bring about a cure for HIV, or at least working along that way. So I'm going to start with a summary for the community, and I hope you can see my slides.
Mm-hmm.
So I think the key challenge with respect to my talk today is going to be how simple I can put this forward to you. So I'm hoping that it's simple enough, but I do understand if it goes beyond and it becomes a little too scientific, but please feel free to ask questions at the end so I can clarify if you have doubts about this. So as a summary for the community, what is the key question of the research we are doing? We're trying to look at whether gene therapy can be rendered safe and practical for widespread use for curing HIV, not restricted to certain populations, but can it be as easy or as safe and practical as antiretroviral drugs are? What are the key findings and take-home message from the talk I'm going to give today? So we are looking at new age genome engineering technologies, and there are delivery platforms for delivering these technologies to specific cells inside the body that are infected with HIV. So the idea is new age genome engineering technologies and cell-targeted delivery platforms can specifically inactivate HIV that's integrated in T cells within the body. And this is where the term in vivo comes in. In vivo means you work inside the body and don't engineer things outside. You try to bring about this change inside the body in the infected cells. So why is this important? The importance is because we are looking towards a highly specific approach that can only target HIV or the T cells that are infected. It enables administration of these treatments via simple intravenous injections. You don't need cell transplant protocols. You don't need cell transplant regimens. And because we're gearing towards simple administration protocols, it's easy to implement. So how is this related to cure? In vivo gene therapy can bring about long-term resistance to HIV and also result in ART-free remission, which is what a cure is all about. And why should we be excited or not about this? We should be excited because in vivo gene therapy can elicit an HIV cure without requiring cell transplant regimens, which has been the current ways of getting of the cures that we have seen. In contrast to other investigational therapeutics, this approach inactivates the integrated HIV provirus, the virus that's residing inside the T cell. It specifically inactivates this provirus without harming the host cell. You don't remove the host cell, but you just kill the HIV inside it. And that's why it's important. And that's why it excites me, at least. So to start with, I'm just starting out with this slide that everybody in this audience will know what is HIV. But for a biologist like me, we look at it from the point of view of the virus. And we know it's a retrovirus, and it causes the acquired immunodeficiency syndrome. So what does HIV actually do? It infects vital immune cells in the body, and these are helper T cells and macrophages. So a key phenotype of HIV that everybody knows is when there's HIV infection, it leads to low levels of CD4 positive T cells. These are helper T cells in the body. And as the name suggests, these T cells help other cells carry out important immune functions. For instance, they help in the production of antibodies. They help in the production of T cell responses to other pathogens. So when these CD4 T cells are killed by HIV, the body is unable to mount an immune response to other infections and therefore gets opportunistic infections that are generally tolerated by the body. And this leads to AIDS. So to just have a gross look at how HIV might look, I mean, this is a pictorial representation, but it's pretty accurate with respect to how it is schematically depicted. The virus itself is circular, like all viruses are, and it has small molecules called the envelope that are present on the surface. There are very few envelope molecules compared to other viruses. This is called the GP120 molecule. What happens if you take a cross-section of the virus? Inside the virus, you have a typical conical shaped capsid And this is typical of HIV and some other retroviruses. And when you cut this further open, you find the RNA of HIV, which is this genetic material. So as each layer of the virus, you take a cross-section and start seeing you would start seeing what's inside, and the RNA is depicted here in blue. So the RNA looks like this schematically. I'm just showing you how it might look. It's got a couple of structures at the very end, and all it does is encode three structural proteins called gag, pol, and envelope that make up the virus. So the virus itself is about 0.1 microns, which is 100 nanometers in size. And to give you a comparison of, or an idea of how big it is, you compare it with the T cell. It is this small, and that's the human T cell. This is the virus that infects the human T cell and brings about all havoc. These videos that I'm showing you are from Janet Iwasa at Utah. She's developed a lot of videos that actually show how the virus replicates, how other viruses replicates in true to live form. And I encourage everybody to really visit her website or look at these videos on YouTube. They're very informative and very simple and really interesting to see. So very briefly take you through the HIV life cycle for you to understand. So this is the virus that I've schematically shown, and the blue dots are the envelope protein that I was referring to. So the virus infects T cells. This is the helper T cell that you can see here. It's called the CD4 T cell. And the reason it infects the CD4 T cell is because the virus binds to this molecule called CD4 that's present on these T cells. That's why they're called CD4 T cells. Now, this is the major receptor for HIV. And the virus binds through its envelope, the blue proteins, to CD4. It also needs to bind to a second receptor or a co-receptor that's called CCR5 or CXCR4. So you need both these being bound by the virus for it to enter the cell. Then the virus fuses with the cell and releases its RNA, the blue depiction of the RNA into the cell. And then the RNA inside the cell undergoes a very special process called reverse transcription. This is very specific to retroviruses. So an enzyme called a reverse transcriptase converts this RNA into a DNA that's shown in red here. So this is one of the few classes of viruses that has got an RNA component, but that gets converted to DNA. And why is this critical? Because most of you will know that human cells and mammalian cells, the genetic material is DNA. And because the RNA now is converted to a DNA, this DNA can enter the nucleus of the cell and integrate into the DNA, and it becomes part of the genetic material of humans. So any human cell that gets infected now has the virus sitting in the DNA as genetic material. This is called the provirus, and essentially becomes it's there for the life of the cell. It cannot be removed. So once it's integrated, of course, the virus, the integrated provirus produces more RNA using the host cell proteins. These RNAs get packaged into new viruses, and by a process called budding, the virus is released into the cell. So the key thing is the entire life cycle here takes about 1.2 days for the virus to enter, get in, integrate into the host genome, and then release more virus particles. But if you look at the clinical course of HIV infection in humans, this is something that many of you might have seen in other talks as well. I'm asking you to look at the two lines. The orange is the plasma viral loads, or the viral RNA that people get tested for in their blood. And the red line shows CD4 T cells. So in the acute phase of infection, very early post-infection, the virus infects, and there's a huge increase in viral RNA loads. It goes up really high. And as the virus replicates and infects more CD4 T cells, it kills these CD4 T cells. So you find a corresponding dip in CD4 T cells as the T cells are lowered. But the body, like it does with any other virus, gains immune control and resists virus, starts producing an immune response to this virus. So for a brief period of time, the viral loads come back down again, and CD4 T cells are stabilized up to a point. But with chronic infection, which can take as much as 1 to 15 years, the virus mutates, escapes the immune response, and the viral loads come back eventually, and this leads to the death of CD4 T cells and AIDS. So I mentioned that there were several steps in the viral life cycle: binding, fusion, reverse transcription, integration, translation, budding. Now, there are antiretroviral drugs that can target each and every one of these processes, really. And that's what forms the arsenal of ARVs that are used in humans. So you have drugs that target each and every stage of this. So you can prevent integration, you can prevent every stage of this, and together that's one of the most effective ways to bring down viral loads. So what happens when you have antiretroviral therapy is the virus can be very successfully controlled. The viral load comes down to, almost non-detectable levels, at least not detectable by the, normal levels or sensitive methods of detection. And CD4 T cells somewhat regain, and you can maintain CD4 T cells for the life of the individual who's been treated with antiretroviral drugs, as long as they take their antiretroviral drugs. But the key is this integrated piece of DNA stays in that infected cell, and this constitutes the HIV reservoir. So this latent provirus and the infected cell remain within the body. The antiretroviral drugs cannot eliminate these. They can only stop the virus from replicating and infecting more cells. So obviously, to cure, it means that you have to get rid of the cell or the provirus that's in the genome. So how do you detect these cells? Now, these cells have only a DNA in them. They don't produce viral antigen. They don't make viral proteins. They don't make viral RNA, at least not something that can be easily detected. So they're pretty much, invisible. So the reservoir, when scientists have estimated how many of these infected cells are in the body, they're really few. So the reservoir is present at a very low frequency. Only about 1 in every million CD4 T cells have this virus when under antiretroviral drugs. So, and these cells that harbor the virus are highly stable. They can exist for long periods of time, long years, in fact, because they become memory T cells. And the estimated half-life of each of these cells with the virus is about 44 months. So under antiretroviral therapy, if you're looking for a natural decay of the HIV reservoir, a natural death of these cells with time, it takes about 70 years to eliminate every infected cell in the body, which is about the lifetime of an individual, which is why people are expected to take antiretroviral drugs for the rest of their lives after they are infected. So this brings us to the question of, why do we need a cure? If you have antiretroviral drugs, why is a cure necessary? So everybody knows that antiretroviral drugs are not the easiest to manage, and there are lots of side effects and lots of other issues for people who are infected with HIV, despite being on suppressive antiretroviral drugs. And essentially, in my view, cure is to improve the long-term health for individuals who are infected with HIV. And second, to really reduce on a community level the transmission of virus to other individuals, because despite antiretroviral therapy, virus can break out, and you can have viral, if there is virus escape, and many times when people are off of antiretroviral drugs, the virus comes back very quickly within a week to 2 months, and this could result in a transmission at the community level. So this is why you try to achieve a cure. And as I reiterated, to achieve a cure, you need to remove the provirus or the virus-infected cell. Now, can a cure be achieved? And this is a wonderful number for me these days, and n equal to 5. For a long time, it was n equal to 1. And yes, when I say n equal to 5, it refers to the number of people that have, potentially be cured of HIV in the past couple of decades. I've listed them, and I really have to take my hats off to people like Timothy A. Brown and Adam Castillejo. So this is Timothy when he visited Yale to speak to the people, and this is my lab with Timothy then. So all these people, were cured of HIV, and when I say they were cured of HIV, it means that there was potentially not a single cell inside the body that contained the provirus, or at least a provirus that was capable of producing active HIV in the absence of antiretroviral drugs. There's also a 6th so-called cure called the viral remission case. It's a Barcelona patient. Nothing much is known about this patient, but there are important differences between how these people achieved their cure and this case of viral remission. In the case of the first 5, most of them underwent conditioning, which is a way of really removing immune cells, which removed virus-infected cells from their body. And then they were transplanted with HLA-matched cells from HLA-matched donors that were negative for CCR5. I did mention CCR5 as a co-receptor. So they were transplanted with cells from donors that lacked CCR5. So there were two things that happened in these individuals. Number one, the conditioning that they went through removed all the infected cells in their body. And number two, when these new cells were introduced, these cells, because they lack CCR5, are intrinsically resistant to HIV infection. So the virus, even if there were cells in the body, the virus cannot spread anymore, so they become resistant to HIV. So the key limitations, however, are this kind of cell transplant that these people underwent is not applicable as a widespread treatment for most HIV individuals. It's toxic, risky. Really, there were other conditions that required a bone marrow transplant in these individuals, which is why they underwent this procedure. Now, the second thing is I talked about the viral remission case. Viral remission case in Barcelona. This patient is slightly different in the sense, as I understand, this person was put on, underwent a supplemental therapy for inducing interferon. I mean, she was treated with interferon, as well as a couple of other cytokines that improved innate immune responses, or particularly macrophage immune responses. So in this individual, the virus-infected cells still remain within our body, but for about 15 years, this person has been off of antiretroviral drugs, but is able to control the virus. And this is something that we call a functional cure. That is, the virus is there in the body, but she's able to maintain or control her viral loads in the absence of antiretroviral drugs. So this is more of a functional cure. So coming back to the cell transplant protocols, I did say that this is not possible to be implemented as a widespread cure approach. So at this point, I just wanted to briefly mention some of the terminologies that are pretty common with respect to cell and gene therapy approaches. What is cell therapy? It is the transplant of intact, living, genetically unmodified cells into a patient to treat disease. The N=5 HIV cures underwent something like this, where they received genetically unmodified cells from another donor into themselves. So with the exception that those cells were already resistant to HIV. Now, gene therapy is the delivery of foreign genetic material into a patient to treat disease. So what's now becoming a major area of interest is a combination of cell and gene therapy approaches. This is because of two things. Number one, it's very difficult to find HLA-matched donors that are CCR5 negative and to undergo transplant protocols. So one way to do this is to take cells out from the subject who requires a cell transplant, modify the cell with foreign genetic material to make them resistant, in this case, for instance, to HIV, by perhaps removing CCR5 or knocking out CCR5, and then reintroducing these modified cells back into the same patient. So it's autologous gene therapy. And clinical trials have so far now mostly sought to transplant the patient's own cells inserted with genes that control or kill HIV. That's where the field is heading towards. But we've not had so much of success with these trials so far, because it appears that you do require a certain level of gene-modified cells, and it's very difficult to get these with a cell transplant procedure, where you have to take cells out from a patient, a certain fraction of cells, treat them and put them back. It's a small fraction of cells. It's not a large fraction. So where do we go from here? The real lesson that we've learned from these cures are, number one, you need to remove the virus. And that can be done in one of two ways. You kill HIV-infected cells, and you remove the proviral DNA. And number two, there's something wrong in the slide, I think. So you need to make the remainder of the cells resistant. So how do you do this? You render the cells resistant by preventing future infection, to prevent future infections with HIV. So the way you do this is you can remove something that HIV needs to make the cells resistant. This is CCR5, or you engineer a robust immune response against HIV, so that even if there are a few infected cells, these cells are prevented from producing HIV and the virus is unable to spread within the body. So I apologize for, there it comes back. So you need to remove the cells and make the target cells resistant, which is what is key. So how can this be done? I mean, there are multiple ways that people have gone around and that many places are researching. One of these is an HIV reservoir-depleting approach, which is, you remove the cells that are infected by with HIV. The approach is called shock and kill. This also happens to be one of the ways. I'm also involved in another Martin Delaney collaboratory called the ERASE that's run out of Emory University. And this is a mainstay. This is the way they are trying to target the virus, as well as several other, research collaboratories. So this is the HIV infected cell where there's a latent HIV. The idea is when people are still under antiretroviral therapy, that means the virus cannot get out or spread to another cell. You shock the cell with a latency reactivating agent. These are small molecules or drugs. It's called the shock treatment. You shock only that cell into producing viral proteins. But the virus is unable to infect other cells because you're still maintaining the whole system under antiretroviral therapy. So in this activated cell, when you have viral proteins being produced, the immune system is now able to, recognize these viral proteins, and you can give immune components like antibodies or T cells that recognize these viral proteins, which leads to killing of the cell, and the cell will die. So the cell is with the infected provirus is removed. So how can you use this approach? Is this something that is viable? It is potentially, but there are several caveats to using only this approach. The shock approach, where you can use drugs to induce HIV only in these T cells, is not very efficient. So far, there's no single LRA that can turn on HIV in all the infected cells. A subset of the infected cells turn on HIV antigens while the others don't. And it's been pretty difficult to find something that can bring about efficient reversal of HIV latency here. Also, the kill mechanism of providing antibodies or using T cells to kill this activated cell is really not optimal. Both of these are something that can target a fraction of the cells, but not the whole. But so this is a shout out, in fact, to the ERASE collaborative that's really trying to improve upon these approaches to get better efficiencies of both shocking only the latent cells and killing only the latent cells, so as to translate it into a cure by killing the infected cell. Now, the second way to do it is it's a silencing approach called the block and lock. So HIV infects CD4 T cells, and this is the cell where it gets latent. So the idea is to block transcription of HIV in these cells so that the HIV is now no longer able to produce more RNA. So there are specific agents that can be used to really, target the HIV provirus and stop it from producing RNA. And in addition, lock the entire HIV in that format so that the virus is always latent. So no matter what you do, whether you give an activating agent or even if you take off antiretroviral drugs, the virus has been somehow modified so that it won't be able to produce any more HIV, any more RNA transcripts or produce more virus. So it remains latent for the life of the individual without the ability to reverse. And this is one of the ways we're tackling the problem in our collaboratory HOPE, where I'm going to be presenting a bit of the data. So the key features of this approach is it does not activate at the infected cell. There is no virus or antigen production. It also does not eliminate the infected cell. And the cured cell kind of remains in the body. It can remain in a latent format, or you can even try and get the HIV proviral genome out. So third approach is, can you target the provirus directly? Now, the provirus is sitting inside the DNA. And this is where genome editing comes into play. This is a term that most of you might have heard. It's all in the news. People talk about CRISPRs all the while. These are all new age enzymes where they do a process called genome editing, where the DNA in an individual, where a copy of a DNA can be inserted. It can be replaced or removed inside the chromosome using artificially engineered nucleases. These are nucleases because they get to the nucleus and work on the nucleic acid. They're able to put in DNA, take out DNA, change the DNA, and these are the new age nucleases that people use. And many kinds that have been studied recently. They range from meganucleases. There are zinc finger nucleases that some of you folks may have heard of. There are talents, and of course, CRISPRs are the flavor of the day. I think flavor of the day now. The way these nucleases act, there's a chromosome. This is the DNA that I have represented as a double helix. So when an engineered nuclease acts on a chromosome, it causes a double-stranded break. The DNA just splits apart. So here, you can introduce new genes, or you can recombine them back, or introduce mutated portions to give you, reconstruct the chromosome or the genome. So this is a DNA-specific double-stranded break, and all these nucleases cause double-stranded breaks to insert, replace, or remove parts of the genome. So when you look at the feasibility of using these, the CRISPR-Cas9 is the most feasible because it's most efficient. It's also highly more specific than these. It's easier to deliver. Efficacy-wise, it's very good. So it's one of the most feasible ways of really targeting anything in a genome in a fairly specific manner. So all these nucleases have been used by people by researchers and scientists in the community to at least, to cut the viral genome of the cell. There are many papers. I've listed a few of them here, particularly CRISPR-Cas9. And I think Dr. Khalili spoke here the last time, and he must have told you quite a bit about this research. And really, they're using very fine ways to cut the proviral genome with scissors. I mean, I know this is exactly what it does. It cuts it out of the infected cell. So the point is, you want to deliver this, and this would come out. But there's a caveat here. To deliver these CRISPR-Cas9s, it needs to find the infected CD4 T cell. It needs to get inside CD4 T cells. The problem here is CD4 T cells, or in fact, all T cells, are extremely resistant to taking up these kinds of viral RNA or proteins. It's not easy to introduce these proteins and genome editing nucleases, or in fact, any DNA or RNA into a T cell. They're one of the most resilient cell types. It's easier to introduce these materials into other cells like skin cells or liver cells. You can use a variety of delivery systems, but T cells are very different, and it's not easy to get into them. And the problem also is, this is the reason, where's Waldo? I mean, where are these infected cells? Because I did mention they're very few and rare. They're 1 in a million. So if you have to introduce any of these to really go and target the provirus that's sitting in the genome of a few cells, where would you even locate them? It's like this. I mean, there would be somewhere here. I mean, you can see Waldo, a few Waldos here, in fact, in this picture, but finding them in the body is almost, it's an impossible approach to do if you have to look at the infected cell, particularly because the infected cell has no markers. I mean, it doesn't produce RNA under antiretroviral therapy. It doesn't produce proteins. How do you know which is an infected cell? Because it looks exactly like the other cell. But Dr. Khalili's group did do this. They have shown in multiple animal models, and in fact, even in humans, in fact, in non-human primates, that it is possible to use CRISPR-Cas9 to go and edit the proviral DNA. And this actually leads to a reduction in the HIV reservoir. So how is this possible? They used a different approach. They used adeno-associated vectors. They used a particular AAV-DJ8 that does not target the infected CD4 T cells. Rather, it targets all cells. The point is, if you can deliver CRISPR-Cas9 to all cells, however, the CRISPR-Cas9 is designed in a manner that it will only target the provirus and won't target any other part of the genome. While delivery occurs to all cells, it will be active only in the cell it is delivered to. So the idea is you really kind of flood the system, and wherever the enzyme goes, if the target is there, it'll cleave out the target. If the target is not there, it won't do anything because it's not going to cut non-specifically everywhere else. That's the idea, because you can design CRISPRs to be highly specific. But and in fact, this has been taken into clinical trials now, and they've actually enrolled in clinical trials. Excision Biotherapeutics really is looking into using AAVs with Cas9s targeting the HIV provirus to get the DNA out of the genome. But what are the challenges to in vivo CRISPR-Cas9, and what should one be aware of? Number one, while the Cas9 can be highly specific. There are risks of having off-target specificity in the sense, while they're designed to go and seek out the provirus specifically, if there is an off-target activity and can cleave somewhere else in the DNA, you would get a double-stranded break elsewhere. So if there's off-target editing within the same cell, you can have nicks in other parts of the chromosome. And of course, those nicks, breaks, double-stranded breaks in other parts of the chromosome. And of course, those breaks can be repaired, and the host cell has excellent repair pathways where you can restore the original chromosome just by looking at it. And that's something that, that can be fairly benign, but this is, however, a risk to be considered. The second thing is, while the virus, as everybody knows, is very variant. Each person's or each patient's HIV quasi-species, and in fact, even within a single patient, there are lots of variations because the virus undergoes mutations. So it's also known that when you use some approach like this, you can have mutations and the virus can become resistant to an approach like this. So there is a risk of off-target editing within the cell, and virus variation complicates things. What else can happen? Second is, I've talked about viral vector delivery. Now, if it's a broadly tropic virus, it can take it to many cell types and not just the infected cell type. So if there are off-target events happening and there are cuts that can happen in other parts of the chromosome, now from within, instead of happening within that specific infected cell, it can be happening in other parts of the body. And this is again a risk. It can get to other cell types and create issues like this, because now you will have these off-target editing happening in other cells as well, besides cells that are infected with the virus. And last but not the least is an immune response. Cas9 as well as the viral vector are immune components. So, with time, the body can develop immune responses to both these. And if the proteins continue to be expressed within the cell, especially Cas9, it can lead to non-specific elimination of those cells which express these. So you really have to shorten the expression time of these proteins within the cell or somehow make them non-immunogenic. That's another caveat. So while this is a great approach, and in fact, many things are being done to reduce all this, these are risks that people have to be considered before really implementing it as a widespread therapy. So at the HOPE Collaboratory that I'm part of, which is run under the leadership of Dr. Melanie Ott, Susanna Valente, and Lishomba Indalawu, we are trying to use this Block-Lock-Excise Approach and in a different manner, and actually trying to look at whether it can be made safe enough for widespread implementation. So I'm going back a little into the Block-Lock-Excise Approach. The block approach is to promote latency by using drugs that can prevent transcription of the integrated proviral genome, so you won't get RNA. And this should potentially act in the absence of antiretroviral drugs. And lock is to permanently keep this provirus in that state, in the sense, even if you take off antiretroviral drugs, even if you take off the block and lock agents, once they've acted on the provirus, the virus now stays latent for life without antiretroviral drugs. And third is to excise or inactivate, which is to inactivate, disable, and excise the already silenced virus. This is the virus that's silenced. Now, why keep the genetic material inside the cell? Is there any way to change this so you can permanently get rid of this virus? Now, we are doing this with a slight difference, and I'm going to focus mostly on this. So we are using genome engineering enzymes, yes, but there are different genome engineering enzymes. The idea is to use nuclease-free genome engineering enzymes. They're not nucleases. They're not going to go and produce double-stranded breaks in the chromosome. They're not going to cut the DNA. What they do is they introduce single nucleotide mutations. So by introducing single nucleotide mutations, the risk is far less because it's highly specific in a particular part where it can be designed to be very, very specific to the HIV Provirus, only at specific areas, and it's a single nucleotide mutation, which can cause harm only to the provirus. And the idea is to have low off-target and high on-target frequencies. So we're looking at two different enzymes here, two different methodologies. One is the CRISPR base editing. This is based on Cas9, but it is a dead Cas9 in the sense the Cas9 has been mutated, so it's not active. but it's combined with another enzyme. I'll very briefly talk about it later. A second enzyme we're looking at is called the BREC1. And this is a recombinase that has been developed specifically for HIV-1 by the collaborators at HOPE. So in addition to looking at the newer genome modification enzymes, we're also looking at more efficient delivery platforms. The idea is to target delivery only to human T cells. It's very difficult to find infected human T cells because they don't differ from other human T cells. But if you have a platform that can target only human T cells, at least you have reduced your target from those billions of cells that are in the body to T cells that matter. So in these, we are using virus-like particles or nanoparticles that have antibodies on them. And these antibodies are targeted towards T cells because they recognize molecules on the surface of T cells and can therefore only get to T cells. And we're using a DNA-free approach to limit foreign protein expression. I did mention that Cas9 is, it's a foreign protein and it can elicit immune responses. So we're looking into delivering Cas9 in the form of protein or mRNA. So you'll have a very short time of expression of these. So it can do the desired effect. And after that, after its activity is done, it no longer remains in the cell. And we're trying to look at delivery platforms with limited or no prior immunogenicity. So the idea is to maximize efficiency and promote a single or a few dose regimen. So coming to the DNA base editor, I did mention it's very similar to Cas9, except that it's a dead Cas9. The Cas9 no longer is active. Instead, it's linked to another enzyme. The Cas9 is just used to lead this enzyme directly to the provirus. So it's highly specific to the provirus. It'll lead it only to the DNA provirus. And what it'll do is convert one single nucleotide. It'll change an AT to a GC base pair. What was originally an AT is to a GC base pair, or it could convert similarly a CG to a TA base pair. Key is it does not have a double-stranded break. It just makes a nick in one part of the genome, which is very, very easy to repair. And we have done this. I mean, this is the part where I show a little bit of data, and I'm hoping, it's just to take a broad take-home message that these kinds of approaches do work, at least in cells, and we are excited about it. If it gets confusing, I can answer questions about it later. So what we have done is we have engineered mutations. Using this, we can bring about single point mutations in several areas of the DNA corresponding to HIV, so that the virus is no longer able to produce this full RNA, and it stops making proteins. And in the absence of, if you introduce mutations at the very end, at the loop regions, you don't get any more RNA produced from the provirus. So when we've tried this in cell culture, you can clearly see, this is infected cells. We have a lot of infected cells here. There's no antiretroviral drugs. There are multiple cells. You introduce these kinds of enzymes, you find that with a single mutation, you can knock down virus by about 2 logs in cell culture. This is measured. We're looking at a log scale, which is about 10,000-fold. 2 logs is from 10 to the 3 to 10 to the 5. You can reduce it up to about 90% completely with many of these mutations. And these are all single point mutations. And the second enzyme I was talking about, BREC1, which is a recombinase, again acts very differently. It recognizes the 2 ends of the proviral genome and recombines the 2 ends and gets rid of the provirus as a circular format. And this is developed by our collaborators, Joachim Hober, Jan Chemnitz, and Ulrika Lange in Germany. So this whole thing is just popped out, not through nuclease action, but by recombining these 2 ends. This is how it comes out. The key thing about this enzyme, that's BREC1, is it's a designer recombinase in the sense it was built specifically for HIV-1. So it doesn't really go or act on any other part of the genome. And it recognizes only the ends of the HIV. It's efficacious on all HIV-1 isolates. It is not immunogenic or toxic. And it has been tested in animals, particularly humanized mice, and in patient cells. And in fact, is now undergoing clinical trials where they're introducing BREC1 into stem cells that are derived from HIV positive donors. This is a cell transplant protocol where cells have been taken out of people living with HIV. They've been treated with these BRCA1 expressing lentiviruses, and they're reintroduced back into the patient to see if you can see some kind of a effect on the provirus or the reservoir, like it was done in the clinical trials. So this is a cell transplant protocol again. But at HOPE now, we are hoping to actually develop this for in vivo gene therapy, where we can use a delivery platform to directly take these BRCA1s inside the body to the T cells where they're required. So how do we do this? So I said that we're looking at T cell targeted delivery platforms. And here I have shown a schematic of one of these. There are multiple kinds we're looking at. These have antibodies on the surface that target this whole virus only to T cells. So you can introduce this virus-like particle. And inside, there absolutely is no DNA. You only have this, any of the proteins. Could be BRCA1, it could be the CRISPR base editing. You can put in multiple of these genome editing proteins alone, which last for a very short time inside the cell and really very efficiently cleared after they do their job. So you have no DNA, they're essentially DNA-free, and they're targeted to T cells, making them highly specific only to a subset of cells where they're required. So I'm again going to show you a little bit of data that we have. So we, as a proof of principle, we decided to use Cas9, the original CRISPR-Cas9, encased in these virus-like particles, but target CCR5 in an attempt to recreate what we saw as cures in these in the 5 cures. So this is actually done in a petri dish. You take a PBMC or peripheral blood mononuclear cells. These are blood cells from individuals. And these blood cells were treated with these virus-like particles, which had, which target CCR5. So these are total blood cells. They have a mixture of T cells, non-T cells, everything inside. And after you treated them, you look for CCR5 in them by running a gel. And this is something called a surveyor nuclease assay. So when you look at the genome, the wild type CCR5 will look like this. And if you've managed to mutate the CCR5, the mutated CCR5 will show up as a different band on the gel. It's a gel picture. So when you look at total PBMC after exposure, we had about 28% of the cells develop mutations. Now, if you take the PBMC out and separate them into T cells and non-T cells, and now look individually at the T cells and not T cells, you find that all the mutation is only in T cells. So you really increased it to about 60% of the cells really getting mutated, which is very relevant, while non-T cells have really 0 mutations. And why is this important? Number one, it can edit T cells. I told you T cells are really difficult to get things into to edit. And number two, it can edit T cells specifically, and it doesn't take them into other cell types. So can you replicate a cure-like approach in an animal model? So for this, we use humanized mice. And I'm going to tell you very briefly about the humanized mouse model so you understand how it might relate to what you have in clinical trials. We are trying this as a preclinical model. So these humanized mice are actually generated with PBMC from HIV positive donors. So these are all blood cells. You put these blood cells inside these mice. These mice don't have their own immune system. They're immunodeficient. But once you transplant these PBMC from HIV positive donors, they develop an immune system that is exactly like, that's very similar to the PBMC from these HIV positive donors. So what you see here is these mice have a little bit of, you can put these on mice on antiretroviral therapy. And these mice are then treated with a placebo or with the VLPs that targets CCR5. And the idea is to then remove antiretroviral drugs and see what happens here, whether the viral load rebounds and what happens to the CD4 T cells when you withdraw antiretroviral drugs. So here, these are first the placebo-treated mice. I'm showing you only the CD4 T cells. The mice have been treated, the mice have been generated with these PBMCs, and they've been treated with placebo that does not target human T cells, and antiretroviral drug has been withdrawn from these mice. So you have an analytical treatment interruption. You see with time, CD4 T cells go down. And why does this happen? Because the virus comes back up. The virus, you look at time, days post antiretroviral drug withdrawal from 0 to 38 days here. So the virus comes back, with the virus which is in red comes back up, it kills the CD4 T cells which are in black, and you find the CD4 T cells go down with time. And as CD4 T cells have gone away, the virus also drops because there are no more CD4 T cells now left in the body of these mice for it to replicate. This is exactly what happens in uncontrolled HIV infection. Now, what happens in mice that have been treated with the VLPs that target CCR5? Find that when after withdrawal of ART, the viral loads do come back, but then they get quickly controlled. But the key is to look at CD4 T cells in these mice. And when you look at it, CD4 T cells initially drop as the viral loads increase, but with time, the CD4 T cells come back to normal while the viral loads continue to drop in red that you see here, which indicates that they're now able to, the CD4 T cells are able to continue growing here. And in fact, we find that we don't find any cells with HIV in these mice after this, which is something like, replicating the cure that we saw in the clinical trials. So this is just to show you that in vivo gene therapy is possible with something like this, and this is where we are looking at. I think I might have overshot my time. I'll stop here. I'll thank all the people in my lab and, of course, all the funding we have from the NIH and the HOPE Collaboratory that have been involved with us in promoting our research. I'll stop on the summary slide, and I will take questions if you have any. I think I overshot, by about 10 minutes.
It's okay. You did a brilliant job of introducing the whole subject. That's the best we've had so far. So we much appreciate that. Now, I think a bunch of us have been trying to answer questions, but here is a good one right here. A problem with this approach is that defective transcripts may still contribute to inflammatory immune activation, from Brad Jones. Are there plans to measure biomarkers of inflammation from these clinical trials?
We have not yet moved that far. So this is being used along with the block-lock-and-excise. So the idea is when we use the block-lock-and-excise, we are hoping that we would completely stop transcription with the block and lock approaches. And this is the final, genome engineering tech that I show is to be used on cells that already have silenced virus. So it's a very good question. Yes, the idea would be to look at inflammatory responses. And again, the caveat also is having an animal model that would portray these inflammatory responses. It's not that easy to do in humanized mice that as you well know. But yes, this is something that we will proceed towards once we get to that stage.
Okay. And Michael, you want to go with your question about only targeting T cells?
Yeah, I just had a question. You were talking about the VLPs, the virus-like particles, only targeting T cells, which just made me think about other cells that may be infected with HIV. They wouldn't be targeted by your VLPs, correct? So a macrophage would not be targeted?
I didn't go into the details, but our VLPs are bound by two different antibodies. You can make them target only CD4 T cells by putting CD4 antibodies or antibodies on them. But the data I actually showed you is with a humanized antibody to CD7 that's present on macrophages as well as T cells. Okay. So quite a few subsets of macrophages have there. The platform I'm showing you can really be, tweaked or reconfigured to go to any cell type based on what antibody you want to use.
Great.
Yeah, that's really good. You mentioned another question. You mentioned resistance. Do you think that the gene therapy process is going to by itself produce resistance as well? Is that what were you referring back to antiviral resistance?
I was looking, CRISPR-Cas9 approaches, there are, when you use genomic RNA to target other areas of the virus, yes, people have demonstrated resistance to this. But the single point mutations that we are choosing, again, that's an excellent question you're asking. These are in areas of the genome that cannot tolerate a single point mutation. So the TAR loop and the splice junctions where GAG to Pol transition needs to occur, at least the way the virus works, a single point mutation there has been proven in any isolate to really kind of destroy the virus. So with this single point mutation approach, we are trying to target only those base pairs that are highly specific and required for the virus to survive.
Richard, does that answer your question? Richard Strange, you still with us? I guess he's gone. Dr. Lee, how about that? Was your question answered about the inflammation markers?
Yeah. Thank you.
Oh, I'm sorry. Richard, you go first.
I just unmuted myself. Yes, thank you for the answer.
Okay, great. Dr. Lee.
Oh, hi, Sugi. That was an excellent talk, Priti. Yeah, no, it was like sort of the first thing that came to mind as you were presenting, and I've been meaning to learn what HOPE is, doing in depth. So that was a great review, but it is a concern. And so I was just curious. It's an easy thing to do, especially the clinical trial you showed that's already getting started, to be able to just measure biomarkers, inflammatory biomarkers from plasma, see what's actually happening. I don't know what sort of in the lab experimentally you can do to see if they're truly silenced or not, but it's like an easy low-hanging fruit from the patient sampling, which would be really informative. It's great if we can, edit and do these interventions, but if we end up still, accelerating aging-associated diseases in this population, it's a problem.
It's a very valid concern. And yes, I mean, for anything to advance into that stage, these are all concerns that we have to be addressing. I mean, no approach really is without risks or, concerns like these. And that's something that definitely has to be looked into from every angle.
Thank you.
Corinne, you had a question as well.
Yeah, let me pull it up. Sorry, I forgot. Can particles of HIV gene recombine to produce a viable virion once they're excised?
So we are not looking at excision at all here. And the way the engineering technologies have so far been used, the simple answer is no. So right now here, we're only looking at introducing point mutations. We're not pulling the whole thing out, or introducing mutations along the existing provirus. So the provirus stays there, but is inactive for life. It just has mutations that will not allow it to replicate or produce transcripts anymore with the interventions we're using. We're not getting it out of there.
Great, thank you so much.
And I thought the most important thing for me, if I got it right, was that with your process, you're not actually cutting, so you're not going to have these crazy off-target things.
Yes. We're only nicking one strand of the DNA to change one nucleotide, and that seals it back. The idea is to not have a double-stranded cut. You don't separate the strands at all.
Right. Wow. I think we have been answering questions as we go in the chat. Does anybody else have any questions that they'd like to ask at this point? I think we got everything. And and Michael has, excuse me, placed an evaluation in the chat. It would really help us if you would complete that. I'd really like to thank Dr. Kumar. Wow, what a great presentation. I mean, you were really the first one to spend some time explaining the basics of this to people, besides the intro section that we had, the session that we had. So this was really helpful. Again, Patricia, thank you for working with Priti to get this right. And everybody else who attended. We'll hopefully see you next Wednesday. What did I say the date of that was?
The 18th.
October the 18th. Dr. Joachim Huber, who is another excise person. He's not from excision, but he's into excising work. And Michael, anything to add? Patricia,
nope.
Richard?
Just putting the chat in the chat again, the evaluation form for today's talk. If you would take a look at that before you sign off, click onto the link so you can get to the form, fill it out for us. We'd appreciate that very much.
And I'm sorry my camera is not working today. For some reason, it won't go on.
I would like to reiterate, if anybody has questions or wants to clarify, feel free to write to me. You have my email. It's my first last name at yale.edu. I'm very happy to, answer questions that you may have, explain things better. I mean, this is the first time giving a community talk here, at least. So I would love to make it more, tangible to the audience.
Mm-hmm.
Thank you for being patient.
Yes.
Thank you.
And there's many thanks in the chat. If you haven't had a chance to see them, many people thank you for today's talk. So
yeah, really excellent.
Feel that.
Thank you so much. Okay, all right. If that we're at 1:02. I love that. Very good.
All right.
All right. Thank you so much, everybody. Hope to see you.
Thank you,
next week.
everybody. Thank you, Priti. Thanks so much.
Thank
Yeah,
you.
very
Thank you for
good.
having me.
All righty. Thanks.
All right.
again.
Take
Bye-bye.
care, everybody.